Hello, I'm Ben Harris. I'm a PhD student at the University of Edinburgh, and today I'm going to talk about the behavior of nitrogen during subduction and what we can learn from in situ analyses of nitrogen in high pressure rocks. So nitrogen is the main component of the atmosphere. It's a key element for life, and the geological nitrogen cycle controls the balance of nitrogen between the mantle and the atmosphere on geological timescales. So nitrogen is degassed at mid-ocean ridges, and it's recycled through subduction zones, where sediments and altered oceanic crust comprise the main reservoirs of nitrogen entering the subduction zone. Some of this nitrogen is lost during dehydration of the slab, and these fluids are incorporated into arc magmas and are either degassed at the volcanic arc back to the atmosphere or incorporated into the crust. But some of the nitrogen is retained in the slab and subducted back to the mantle. And the current understanding of how the nitrogen in the atmosphere has evolved over time is not very well constrained. So one of the ways we can look at this is to understand how nitrogen behaves in subduction zones and therefore estimate the flux of nitrogen through subduction zones. So nitrogen in rocks is mainly hosted as ammonium, which substitutes for potassium. And correlations between bulk nitrogen and bulk potassium contents suggest that micas are the main host of nitrogen in sedimentary rocks and in some mafic rocks where micas are present. So previous studies on nitrogen have been limited to whole rock analyses, and relatively little is known about nitrogen mineral fluid partitioning during subduction metamorphism, which controls how easily nitrogen is lost into fluids and therefore lost from the slab. The studies on nitrogen contents of subduction zone rocks show fairly mixed results when compared to their unmetamorphosed protoliths. So there's a need for more studies to understand how nitrogen behaves during fluid rock interaction and how that affects the nitrogen loss in subduction zones. So the aims of this study are to analyze nitrogen in situ by ion microprobe in minerals from high pressure rocks. This is a new technique for analyzing nitrogen, and it will allow us to compare nitrogen to other fluid mobile elements, which we can analyze in the same run, and to look at textural controls on the nitrogen distribution. We can use this data then to explore the fluid rock partitioning behavior of nitrogen in natural samples and compare this to experimental data on mineral fluid partition coefficients. And we can say something about the nitrogen loss during subduction. So we will look at two case studies, one from the Western Alps and one from Ecuador. So this is a new technique for analyzing nitrogen. It's not been tried before because nitrogen doesn't ionize particularly well on the ion probe. However, we now have a good routine where we can get good signal and we can distinguish between different interferences. So the main interferences on nitrogen are silicon and carbon. And we can see here in our phengite primary standard, we have a clear nitrogen peak, which is distinct from the silicon and the carbon. And in the pyroxene, we have no nitrogen. So we have a phengite primary standard, which was analyzed by F2AR for nitrogen contents. And we have a muscovite secondary standard, which we were able to reproduce relatively well. So the first case study we're going to look at is from Lago di Cignana in the Western Alps, the classic ultra high pressure oceanic crust locality. And this is a garnet-phengite quartzite, a meta-chert, which contains garnet, quartz, phengite, and rutile. This was previously studied for its boron isotope composition. So it displays a large range of boron contents in phengite and a large range of boron isotope values. And these were interpreted to reflect fluid rock interaction at peak conditions with an isotopically heavy fluid, which was rich in boron, and interpreted to reflect a serpentinite source. So we analyzed nitrogen in these same phengites. We found that nitrogen doesn't show any correlations with the major element composition of the phengite, for example, the silicon. But we do see correlations between the boron, lithium, and nitrogen trace elements. So we modeled this data in order to constrain the phengite fluid partitioning behavior of nitrogen. We use an open system fluid rock interaction model where the concentration in the mica depends on the concentration in the fluid, the initial concentration, the fluid rock ratio, and the phengite fluid partitioning coefficient for nitrogen. And in order to constrain the composition of the fluid, we know that serpentinites in bulk rock contain very low nitrogen contents and low lithium contents. So serpentinite derived fluids should also contain low nitrogen and lithium contents. So we used the same boron compositions as we used in the boron isotope modeling. We used lithium partitioning data from previous studies and a low lithium concentration, a 0 lithium in the fluid. We also assumed no nitrogen in the fluid, and then we varied the partition coefficient for nitrogen in order to vary the slope of these models and fit the data that we observe. In particular, this positive correlation between nitrogen and lithium. And in order to fit the slope of this array, you need a phengite fluid partition coefficient, which is less than about 1.5. And this also produces similar fluid rock ratios to the boron isotope modeling, which is nice and consistent. So there is some experimental data on phengite fluid partitioning. It's very variable. It comes from quite a wide range of experimental conditions, and R values of less than 1.5 fall into the lower end of these range. Importantly, quite a few previous models of the subduction nitrogen cycle have assumed values around 10 based on data through rubidium, which has a similar ionic radius to ammonium and was suggested to behave similarly. But our data suggests that nitrogen is more fluid mobile and is therefore more easily lost from the slab. So the second case study that I want to look at is from the Raspas Complex in Ecuador. This is a blueschist, which has similar peak temperatures to the Cignana sample, somewhat lower pressures. And it contains glaucophane, a bit of garnet, epidote, phengite, paragonite, and some carbonate. So we analyzed nitrogen in the minerals from this sample. The main nitrogen hosts are the white micas, phengite, and paragonite. The glaucophane and the epidote both contain less than 5 ppm nitrogen, so they're insignificant as a host of nitrogen in this sample. And we see in the phengite a positive correlation between the silicon content of the phengite and the nitrogen. Similarly, we see positive correlation between silicon and boron and a negative correlation between silicon and lithium. So the simple explanation for these trends would be that nitrogen and boron was lost during retrograde fluid interaction, where the silicon content of phengite decreased and the lithium was added. But this would require a fluid which is very rich in lithium, poor in nitrogen and boron, and there doesn't really have an obvious candidate source for these type of fluids in subduction zones. So alternatively, we tried to think about explanations where the trace elements are redistributed amongst different phases during fluid rock interaction, particularly the paragonite and the phengite. So one of the curious features of this sample is that it contains a very high paragonite mode, about 15%, also about 15% phengite, and it contains carbonate. So we conducted some pseudosection modeling to explore the influence of adding carbonate to this system. And this is a section of pressure against the mole fraction of carbonate in the accompanying fluid. And we see that at the peak pressure temperature conditions for Raspas, which is about 550 degrees and about 1.5 to 2 GPa, this sample is predicted to contain about 20% omphacite and little to no paragonite. However, in the presence of a carbonate-bearing fluid, the assemblage which we see in the rock is stable. And in this field here, as you go from left to right across this field, the paragonite mode starts out at 0, increases to about 25%. So the 15% we observe is about in the middle, and the silicon content in the phengite decreases, and the glaucophane mode also decreases as glaucophane is consumed to form paragonite and carbonates. So we can now think about the trace element budgets in terms of these new phases. So the correlations that we see in the phengite can be explained by partial reaccumulation of the phengite with new phases growing during fluid rock interaction. So the growth of paragonite sequesters nitrogen and boron from the phengite, and therefore we see a decrease in the silicon content during fluid rock interaction that's associated with a decrease in nitrogen and a decrease in boron. However, the glaucophane that we measured was quite rich in lithium, richer than the phengite and the paragonite. and the glaucopane breaks down to form paragonite during fluid rock interaction. And so this can release lithium and explain the opposite trend that we see in the lithium data in the phengite. So we estimated the budget of nitrogen during the fluid rock interaction by taking the phengite with the highest silicon content to represent the initial nitrogen composition, and then the mean phengite and paragonite nitrogen contents to represent the final composition. And then using the mineral modes, we reconstructed the bulk nitrogen content before and after fluid rock interaction. And we suggest that there is little change in the nitrogen content during fluid rock interaction in this sample, unlike what we observed in Cignana. So what are our conclusions from this study? Well, firstly, that in situ analyses of nitrogen by ion probe are possible and they can give useful data. We conclude that nitrogen is moderately fluid mobile, more fluid mobile than has been assumed by some previous studies. The stability of nitrogen-bearing phases during fluid rock interaction exerts a strong control on the rock fluid nitrogen partitioning, and therefore nitrogen is a complex element and its variable partitioning behavior should be considered in models of the subduction zone nitrogen cycle. So now in future, we want to look at a more coherent set of samples across different metamorphic grades to get a better handle on the mineralogical controls on nitrogen loss during subduction. So thank you, and hopefully there are some time for some questions.
Thanks for a great talk. Yeah, so we definitely have we have 3 minutes for questions, and then we have a coffee break coming up. So by all means, if there's any questions, please ask away. I don't see any questions in the chat yet, but if you could talk a little bit about the uncertainties on your nitrogen measurements using this new SIMS technique. So you showed a very small difference at the end there, 57, 59. I mean, what are we talking about here?
Yeah, so we haven't really yet done anything to optimize the method for doing this. So there's definitely work that could be done there. But I mean, we have one phengite secondary standard which we used to calibrate our analyses, and that seemed to be reproduced reasonably well. But having said that, these phengites that we have, which are measured by FTIR, are not super homogeneous. So yeah, there is definitely more work that could be done on getting the uncertainties down and getting the method done better. But we were really just trying to see whether it's possible and whether it does give some data that appears to be useful. But yeah, for example, the bulk rock calculations that I showed at the end with the 37 versus the 39, yeah, they're not distinguishable. Like that's with an uncertainty.
That's really interesting stuff. I mean, again, there's what do you think is the big difference why you were seeing a change in the one example and not seeing it in the other example?
I guess there's a couple of reasons. Well, firstly, just that in the first example, the only nitrogen-bearing phase we have is phengite, and it doesn't really appear to have recrystallized extensively during the fluid rock interaction. So there's no like redistribution of nitrogen. So you're just partitioning nitrogen between phengite and the fluid. Whereas in the second example, we're growing a new nitrogen-bearing phase during the fluid rock interaction, which is the paragonites. And so that seems to be taking in the nitrogen that's instead of it being lost to the fluid. Okay. But also, I mean, another thing with nitrogen is that it's a redox sensitive element, like, and that almost certainly affects the partitioning behavior. So yeah, there are lots of things which remain to be explored about that.
Okay. That was a wonderful talk. I quite enjoyed that.